Start the live stream here. Got it. And got it. Okay, and I'm going to record the Zoom. One more thing I'm going to do here. We are in the cloud. Excellent. Welcome everyone to the second weekly Water, Wetlands, and Watershed Seminar. We're kind of bootlegging things because the projector broke, but we're very happy to have Dr. Joshua Patterson with us today. Let me see. I'm going to put this camera over there, and we'll see Christine. And I'm going to have the introduction today by Christine. We'll have Josh. Are you on the Zoom call?
Yes.
You're on the Zoom call, and you'll need to share your screen.
Oh, with the Zoom call.
Yes, so that the people on Zoom can see, and everyone in the room is good, and then there's YouTubers also. So maybe while he's doing that.
Okay, sure.
Intro.
I'll jump in and do an intro just to keep everything moving along. Hi, everyone. It's my great pleasure to introduce Dr. Josh Patterson today. Josh joined the School of Forestry or Forest, Fisheries, and Geomatics Sciences here at the University of Florida in 2014. You're in my cohort, Josh. His research and extension programs are focused on restoration aquaculture, and he's housed at the Florida Aquarium's Center for Conservation that's in the Tampa Bay area. It's a beautiful facility. He's got an amazing lab and a really rich collaboration that's there. So if you're interested in these topics, I encourage you all to go visit him there because it's quite a setup. Restoration aquaculture, we're going to hear about what this is today, but the areas of exploration and outreach in his lab focus primarily on the use of aquaculture in coral reef restoration, including the use of corals, sponges, and sea urchins, as well as the restoration and protection of seagrasses, bay scallop population enhancement, and various fisheries aspects of large-scale habitat restoration. Josh works extensively with a variety of stakeholder groups, ranging from Florida Fish and Wildlife Conservation Commission, NOAA, various nonprofit organizations, and private businesses in this effort. So today we're really excited to hear his overview of his program on restoration aquaculture. So please join me in welcoming Josh too.
Yeah.
Thank you so much, Christine, for that introduction, and thanks for the invitation to come present. So we appreciate it. Without further ado, Dave, thanks.
Yeah, sorry, man. I know we got you awkwardly set up here and everything like that.
That's all. Oh, I
Yeah.
understand it.
Yeah.
Okay. All right, cool. Thanks. So, yeah, as Christine mentioned, this is going to be sort of an overview of restoration aquaculture. Probably could have called it Restoration Aquaculture 101. And it's sort of a new and maybe emerging field in terms of pulling all of these disparate activities together under this one umbrella. And so we're just going to kind of walk through restoration aquaculture.
Probably because you got to click back on. There you go.
All right, cool. Very broad outline of sort of three segments that I hope to get to today. So the first is some definitions and also examples of what is restoration aquaculture. And then some considerations for sort of practice or practitioners when you're doing restoration aquaculture. What are some things you want to be aware of, or as you're sort of thinking about the topic? What are some things you want to be aware of? And then if I don't ramble on too long on the first couple, we will try to get to some just research examples of stuff that we've been working on in my lab. And the questions that are there, there's bunches of them, the questions that need to be addressed for this field. So in terms of definitions, and these are going to be some terms that you've probably heard before. A stock enhancement is certainly a form of restoration aquaculture. The photograph here is a salmon hatchery in Japan. The orange things in the tubs there are actually all salmon eggs. So this gives you an idea of the scale of this sort of aquaculture. And the statistic that I have is in Japan, 9 out of 10 chum salmon that are harvested by commercial fishers come from stock enhancement. So they're stocked. It's almost, you can think of it as like a put and take sort of situation. So the term stock also sort of brings to mind like a commercially or recreationally harvested species. It doesn't have to be a finfish, could be a bivalve, something like that. So there's a specific stock that we like and we would like to enhance it. And so we can sometimes do that through aquaculture. So that's one example. Another example is in terms of threatened species. So I did my master's work at Kentucky State University in Frankfort, Kentucky. And the most imperiled group of animals in the state of Kentucky are freshwater mussels. So the Kentucky Department of Wildlife and Fisheries actually runs a mussel hatchery. It's really fascinating from an aquaculture perspective. They have this crazy life cycle. where they'll insist on the gills of specific fish. So not only do you have to have the mussels to grow them, you have to have the fish, and then they drop off. And so it's a really neat sort of, from just a pure aquaculture perspective, sort of program there. So not just mussels, there's corals, which is another sort of overlaps with habitat that I'll talk about in a second, but there's a lot of examples where it's not something that we necessarily want to harvest, it's not a stock, but it's just a threatened species that for biodiversity or other reasons we'd like to have around. And then the final one here is, so if you think about freshwater mussels, that they're a species that sort of resides within a habitat, right? They don't create habitat themselves. This last example is oyster reef restoration. This photo comes from the Chesapeake Bay. And so restoration aquaculture can also often be used to restore habitats. So sometimes with oysters, that can be like a single species where you're able to recover the different structures and things that are part of an oyster reef. Some of that biodiversity will come around it. But that's just sort of one example, and corals is another one, as I mentioned, that sort of overlaps with threatened species. So thinking about things from the ecological perspective, sort of shifting away from aquaculture, there's a spectrum of approaches. And so I've been using the term restoration aquaculture. It was part of the job title when I was hired into the position, so that's probably one reason. But also there is, one of the leading journals in the field is Restoration Ecology, which was published by the Society for Ecological Restoration. So this term restoration is actually used quite a bit. In reality, it's probably more of like an aspirational term than what we in practice end up doing. And so this figure sort of depicts that. And I apologize, I don't have a reference for this figure. I tried to find where it came from, did like a reverse Google image search and wasn't able to find it. But I think it's sort of a good depiction of what the ways you can think about this. So on the x-axis is diversity and structure of a given ecosystem, and on the y is ecosystem function. And the goal is generally some reference ecosystem, right? When we think about trying to restore something, we would like to bring it all the way back to some historical reference ecosystem, some baseline. And what that baseline is can be debated and sort of what that time point should be. But if we were able to actually do that, that would be fully, truly restoration, right? A step back from that is rehabilitation, which is restoring some part or bringing some part of an ecosystem back. And then even further down the line is enhancement. So think back to stock enhancement, right? So we've got this one particular species or stock that we'd like to use for a specific purpose. And so we would like to try to enhance that. And this goes for any form of sort of restoration ecology. It can be done terrestrial as well as aquatic systems. And then the yellow dots there are replacement. And so the concept of replacement is sort of goes outside of the reference ecosystem. So it's creation of a new functioning ecosystem on a site where it possibly didn't historically exist. So if we think about in Florida, we get a lot of these areas that used to be sort of mangrove swamp and were channelized for various reasons. We might install a living shoreline there with some oyster reef restoration and things like that. That would be replacement technically rather than fully restoration, right? Because that area wasn't necessarily historically, and again, what time period we're talking about, but that area might not have historically been oyster reef. Okay, and so now pop quiz. We're going to move into, in thinking about this, I find it helpful to just use examples of things that happen in the real world and then ponder whether or not that would qualify as restoration aquaculture in my book, right? Because that is the job that I was hired to do. So there are no right or wrong answers for these. I'm going to give you my opinions, but I want to see sort of what you all think first, right? So this is one that I found that's actually really interesting. It was a USDA program for old fish production ponds in Michigan. And the idea was to seed them with emergent wetland vegetation and create wetlands, basically, in these areas that had been basically fallow aquaculture fields, if you want to think about them like that. So do you guys think that qualifies as restoration aquaculture?
No.
No, I love it. Well, I would actually say yes.
Well, what was it before it was that drained? I mean, it's probably a drained wetland or something, right?
So it could be right if it was a drained wetland, then it is restoration, right? It's not
restoration of aquaculture.
So that's the point that I'm glad you said no, because that's the point that I wanted to get to here is that I would consider emergent wetland vegetation aquaculture, right? So it's an example so where do you draw the line, right?
Does it have to be an animal? I guess it's.
Surely, yeah. So it definitely doesn't have to be an animal, in my opinion.
Okay.
We consider alligators aquaculture. There's an alligator aquaculture industry in Florida and Louisiana as well. And so is that a fully aquatic animal? The other example I think of is in terms of mangroves, right? I would love to consider red mangroves. If you're and there are native plant nurseries that culture red mangroves and do mangrove restoration, that to me would be aquaculture. But a white mangrove or a buttonwood that's a couple steps into the terrestrial sphere, right? Is that still aquaculture? So where exactly you draw that line? I don't know, but to me,
That's going to be on the test, okay?
that's an essay question.
Right. It's pass fail, there's no test, Okay, go ahead.
Make your argument.
Yeah.
So anyway, just sort of trying to draw that aquaculture concept out as broadly as possible. It gives me more areas to look at. So this one's a little maybe tongue in cheek. I don't know if y'all are familiar. This is Osborne Reef off the coast of Fort Lauderdale. So this was a couple million tires that were installed in the 70s to create structural complexity and potential fish habitat and that sort of stuff. Has turned into a bit of an environmental disaster. The tires have broken loose. And it's, I think, over 20 years now that we're still working to clean these tires up. But the concept I want to get across is that of just artificial reefs. Artificial reefs are a big deal in Florida. And when they're installed strictly as fish habitat or as dive attractions, do you guys think that qualifies as restoration aquaculture? No, all right. I don't think so either. Because there is no aquaculture component to that, right? We're modifying the environment sort of with the intent of attracting or augmenting populations, but there isn't necessarily that sort of intentional growing of the organism, I guess. This is a more maybe modern, more positive example. I think this was in 2009. The Vandenberg was sunk off the coast of Key West. And actually, and I'm going to talk about later the intention of why you're doing these things, right? So one of the stated intentions when this was done was to draw dive pressure away from local reefs. And so there is a conservation restoration type angle there, right? Where we're hoping that by installing this artificial reef, we're going to, in some sense, conserve, natural reefs. We've already sort of talked about this one. So in my opinion, this one is not restoration aquaculture because again, it lacks that sort of aquaculture component. Okay, here's another story. So this one's from Thailand. There was a reservoir construction project on this island. Massive erosion and landslides buried the adjacent reef in about 2 meters of sediment. So part of the idea, one of the things that was thought about was bringing in these artificial structures and then actually taking corals and gluing them down to the surfaces and outplanting corals to those surfaces. So same, right, artificial reef concept, we do this quite a bit. But now we're actually intending to grow an animal and actively plant it. So is that restoration aquaculture? I think so. Yeah. So to me, yes. So that would be an example of restoration aquaculture. So here's one that's even maybe another step removed from the active culture of the organism, right? And this is an example from Tampa Bay Watch. On the west coast of Florida, we're generally sort of not recruitment limited when it comes to oyster habitat. There's plenty of oyster larvae floating around in the water. So if we can get some marine-friendly concrete structures like this, put them in the water in the right places, and there's a lot of work that goes into what those places are, oyster larvae will generally settle on them and grow. So again, artificial reefs with the intent of culturing something relatively actively, although we're not like, seeding the oysters on there. So how about that one?
No. Maybe.
I want to say yes. Please, yeah. I would like to say yes, just because this is a huge area of work in Florida. And to me, it goes far enough in terms of actively modifying the environment. If a bunch of barnacles encrusted on that thing, you would be really bummed out and you would try to figure out how to do it so that you got oysters the next time. So the intent, I guess, is there enough for me. But I totally get it if you say no. All right, to flip it a little bit more. So here's larval bivalve seeding, right? So this is on the east coast of Florida, where there maybe aren't as many oyster larvae floating around in the water. So this is a bottle full of oyster larvae that were produced in a hatchery, and they're being released into the environment with the intent that they settle and grow. So how about this one? Restoration aquaculture?
Yes.
Cool. Yes, you've got a really intensive hatchery phase. You're intentionally spawning these oysters and then releasing the larvae. What happens to them after that is sort of the question in that particular instance. Here's another one, caging bay scallops, similar to, there's a couple of different ways this can be done, right? So I've seen it where bay scallops are produced in a hatchery. They're given to citizen scientist volunteers, and they are sort of grown out in cages hanging under docks with the intent of restocking wild populations. I'll just tell you, to me, that would be restoration aquaculture. You've got the hatchery phase, you've got the people intentionally growing bay scallops and then releasing them to augment wild populations. FWC has a big program up in the Panhandle, and this has been done prior even in Tampa Bay area, where we're going and collecting wild bay scallops and caging them as to intentionally create spawning aggregation, right? So to bring the animals closer together such that when they spawn, we've got larvae going out into the water. That second one, would that be restoration aquaculture to you all? I see, good, cool, all right. I see some yeses and some no's. I have a maybe. I don't even know. I haven't fully decided on that one yet. It's maybe a little too far removed, I think, for me. But I could be convinced. All right, just a few more. So I don't know if y'all have heard of the concept of fertilizing the ocean with iron. It has a couple of conservation angles. In many areas of the open ocean, iron is a limiting nutrient for primary productivity. So if you dump a bunch of iron in the ocean, you get a phytoplankton bloom. Those phytoplankton consume carbon dioxide, carbon. They can sink and die and sink to the bottom, where that carbon could be sequestered for a long time. So there is a conservation climate change sort of angle. There's also been the thought that this might improve fisheries yields by improving primary productivity and on up the food web till you get to some fisheries of like human interest. So how about this one? We're trying to grow phytoplankton. Is that restoration aquaculture? Okay, cool. No, I don't think that one is. So, all right. This is aquaculture, right? I know aquaculture when I see it. This is very control intensive form of aquaculture. We're probably monoculturing specific algae strains with the intent of selecting for those with high, some of you engineers probably know better than me, compounds that are useful for biofuels. Again, there's a conservation restoration angle here, right? An environmental restoration angle of moving away from fossil fuels. And it's definitely aquaculture. But is this restoration aquaculture, you think? Marks, yeah, we're good. No, again, just a little too indirect, right? It's a little too indirect from directly restoring a habitat or a species. All right, two more. Sediment tubes and seagrass propeller scars. If you're not familiar with propeller scars, we've done some work on this particular issue. When boats go through water that is too shallow, they can cut this trench, and then the seagrasses that we have here in Florida will not grow back down into that trench if it's deep enough. So these essentially become permanent. You can see bunches of them on the earth. Erosion can occur, and the scar can even get worse. It's a pretty low-tech solution that just involves filling a cotton tube with sediment and basically bringing it back up to grade. The cotton will biodegrade, and the seagrass will grow back over it. So is that restoration aquaculture? I might even by saying that I've worked on it. In my opinion, yes. So it's similar to the oyster ball, right? That is intended to be colonized by oysters. You're modifying the environment with the active intent of something growing into that. And then the last one is another sort of seagrass restoration technique that I find really cool. That as in a lot of areas, seagrasses can be nutrient limited. And so there's been a good number of papers and stuff that show if you put out these bird stakes in the right areas and leave them out for the right amount of time, the added nutrients that come from the birds will enhance the seagrass growth. So what do you think about this one?
Yeah, if you're gonna say the other one was. Yeah, no.
Maybe, so this maybe should have been a maybe, but I sort of, like Mark. Yeah, I sort of share that. It's maybe not quite of a direct like environmental manipulation enough.
I mean, fertilizer. I mean, planting for anyway. But please.
Yeah. Well, also right, like fertilizing the ocean with iron.
Yeah.
It's not. So this is just fertilizing seagrass. So
True.
It's a little closer than fertilizing the ocean with iron. All right. So those are some definitions. So hopefully now, at least when I say restoration aquaculture, you have some sort of concept of what I'm thinking about. So considerations for when we do restoration aquaculture, and I usually lead a talk like this with this sort of information, but just wanted to sort of set it around a little bit. So in my opinion, it's really important to draw a line between conservation and restoration, right? And so we don't want the ability to grow something and try to put it back to be an excuse for failure to conserve on the front end, right? So restoration aquaculture doesn't always work, and it can be expensive. And so it's a great tool to have in the box again, which is the title of the talk, but we don't want it to be an excuse for not conserving. And so coming along with that is acknowledging the limitations, right? So restoration aquaculture really appeals to that part of our human nature that wants to do something now, right? This thing used to be there and I liked it and I want it back and I can grow it. And so I'm going to grow it and put it back and I feel good that it's back there. And I get that. And so, but I do think that at times restoration aquaculture has particularly been rightly criticized for maybe overselling the sort of restorative ability of the practice. So we do need to acknowledge limitations. And along with that is talking about the other tools that are in the box for managing aquatic systems, right? So dealing with climate change, dealing with land-based pollution, all of those other things that are very important in terms of getting habitats back to historical baselines. And so I've already sort of touched on this at this point, but it is just one tool in a large box. And so that's the end of the negativity. Now we're just going to talk. So keep those things in mind, of course, when you read about restoration aquaculture type stuff and see it in the news. But there are a lot of good aspects of it that are worthwhile. So I do think it is something that's worth pursuing. All right. So from the sort of philosophical, theoretical to very practical, when we're going to do restoration aquaculture, permitting is something that is sort of unavoidable, right? And I guess maybe this is what I think of when I hear the word permitting. This is maybe what comes into a lot of people's mind. And permitting can certainly be onerous and in some cases almost prohibitive. However, what I would say though is think back to the Osborne Reef example, right? You could never permit that today, and rightfully so. So permitting is there for a reason. It's you know, there are certainly ways we could try to streamline it. And in fact, we're doing a whole workshop about that in about a month, permitting and policy and that sort of thing. But it is something that is, you know, sort of important. So this is just sort of a list of agencies with potential jurisdiction if you were going to try to do restoration aquaculture work. And I'll walk through them and just sort of give examples. The U.S. Army Corps of Engineers has jurisdiction over anything that goes into navigable waters. And so an example is the seagrass propeller scar sediment tubes that I talked about. Those are actually considered fill and have to be permitted by the U.S. Army Corps of Engineers. The NOAA and U.S. Fish and Wildlife Service have jurisdiction over a lot of threatened species. So endangered freshwater mussels would be Fish and Wildlife Service. NOAA tends to take things that are in the marine realm. And another example is for Florida Keys National Marine Sanctuary. It's sort of a boundary down in the Keys that if you're going to be doing work, especially restoration aquaculture type work, it's almost certainly going to require a permit from the Keys Sanctuary. So we work with NOAA and Fish and Wildlife a lot. And then at the state level, there's FWC. So any cultured animal that you're going to be releasing into the waters of the state of Florida is going to require a special activity license through FWC. Florida Department of Environmental Protection has jurisdiction over something we call sovereign submerged lands that belong to the state of Florida. So we get a lot of DEP permits. And then the Department of Agriculture and Consumer Services, in terms of it is still aquaculture, right? And especially on that production end, your facilities are going to have follow these best management practices and other things and be sort of licensed by FDACs. So those are just some of the areas. And so any given project might have permits from 2 to 5 of these agencies. All right, and finally, and this is something that also doesn't apply just to restoration aquaculture, but I think it's really important to have goals and objectives lined out when you begin your project. And I'm not going to sit here and tell you that every project I've ever done has, you know, 100% met the SMART objective acronym, but you know, it's a good thing to keep in mind and to try to do. So the S is for specific. You want to know exactly what it is that you're trying to do, right? What is the goal? Is it, you know, enhancing population of a certain species? Is it increasing the percent coverage of seagrass? What is your metric? And along with that is measurable, right? Your metric needs to be measurable. There should be some data that you can collect that support whether or not you met your specific objective. Attainable is a really important one. If you set your goals too high, your failure rate will increase, right? And so people will begin to not believe that you can do what you say you're going to do. However, you also want them to be, you don't want to set your bar too low, right? Because then you won't achieve as much as you potentially could have. Relevant is also important, and I feel like it's often overlooked. So you want to increase the population of that species. That's a great specific objective, but why? right? What is the ultimate? Are you trying to improve water quality? Is there habitat you're trying to create? Are you trying to expand a recreational fishery? Those sorts of things. So what is the relevance of what you're trying to do? And then making it time-based just always helps, at least in my head, having sort of a finish line that you're at least striving for, whether or not you actually do it. So those are some important things in terms of setting your goals and objectives. Cool. Where's your research examples?
Yeah, you got 20 minutes, so you're good.
Oh, nice. All right, good. Cool. All right. So I've got a couple of research examples. These were sort of selected. I apologize off the top. I'm going to leave out a lot of important background and context, as well as experimental design and stuff. It's all relevant, and just sort of hit the high notes. But I do have some citations and stuff in there. So if you're interested, most of these are published, and you can check them out later in a lot more detail if you want. As I mentioned also off the beginning, it's a really cool field to work in because there are endless research questions surrounding restoration aquaculture. A lot of these activities are relatively novel, and so there's just a lot of things that are sort of unknown. So this first one, Christine mentioned in my introduction that a lot of probably the greatest proportion of our work has been in coral culture and outplanting. So we study both the ways that we grow coral and the ways that it can be put back on the reef most effectively. And so this has been a pretty active area of research. I've had a couple of PhD students, a couple of master's students, and many different partners over the years that I've been doing this. And so apologies to most of them. I'm just going to focus on Joe Henry, who's a PhD student who finished just this past summer. And I'm going to focus on a couple of his dissertation chapters and publications. And the other partners that were involved in this particular research were the Florida Aquarium, who houses me. And then Joe was able to get some funding from the University of Florida Foundation through the Cynthia Melnick endowment to help with the outplanting, the second sort of area of this. So the first part, and this was designed in partnership with the Florida Aquarium, corals can be propagated in a couple of different ways. So they can be asexually fragmented, and that is the way that most biomass is produced for restoration to go back to the reef. It's really great, it's really efficient, it can be done in ocean-based nurseries, but one of the issues with it is you're sort of stuck with the amount of genetic diversity that you had when you initially brought those corals into culture. Corals can also reproduce sexually by spawning, releasing sperm and egg, which turns into a little larvae that finds a hard place to settle and can grow into another coral. The cool thing about that is it creates a new genotype, right? It creates a new genetic individual for every little coral that settles is a new recombination of genes that didn't exist. And so there's a lot of interest in potentially trying to select for corals that have traits that might be more resilient in the face of future challenges and that sort of thing. So way down in the weeds from all of that is this study that Joe ran looking at ways to improve the efficiency of ex situ sexual propagation of corals. This is something we do a lot in aquaculture. We have a target species that we'd like to grow. And so we're just working on figuring out ways to improve that process. And one of the ideas is the use of these grazing herbivores. This is not our idea. The Florida Aquarium had snails and other things in their tanks long before we started doing this research. But trying to select which ones and the appropriate densities is something that we were interested in. So we set up a pretty straightforward experiment. The snail on the left is Batellaria minima, and on the right is Lithopluma americanum. These are both native snails, which is really important when you're doing this sort of work. So the facility in Apollo Beach is completely biosecure for really keys, not even Caribbean wide. So when you think about reefs, you think about Caribbean and Pacific reefs. Pretty much every organism and everything that we have in those greenhouses has come from the Florida Keys, because that's where most of the corals are sort of destined to return to. And we're really conscious of not trying to bring in zooxanthellae or other things that could become problematic when you get back in the environment. So that's an example of what one of the replicate tanks looked like. We had 10 tile subsamples basically within each tank, and a common density of both grazer species as well as control of those snails. The little dots on those tiles are coral recruits. So we went through really carefully and selected tiles that had 3 to 4 staghorn coral. Acroporesorda cornis is probably the species that has been used most in active reef restoration in the past. And the species profile is really diversifying now, but it's still a species of considerable interest. And not going to bore you with graphs on this one. So these are actually sort of representative photos of individual subsample tile replicates from the experiment. We found that Lithopoma americanum actually improved the growth and survival of these coral recruits better than the other snail, and actually obviously better than the control as well. We recorded about 75% survival of the recruits to 126 days, which was to date, I think, the highest survival for this particular species in ex situ culture. So that was pretty cool. One of the ways that was achieved, and this is why the interest was there to do the study, is that one of the things that the Florida Aquarium does is hand cleans these tiles. So with dental instruments, it's extremely labor intensive. But it really helps, because otherwise you just get algae. You try to manage your water quality and stuff, but you still get this algal overgrowth. So these photos were taken every 3 weeks. When we collected data, these tiles were all hand cleaned, and we really rigorously timed the amount of labor that went into cleaning these tiles. So in addition to improving growth and survival, the addition of the snails cut the labor time by more than half. So following this research, the aquarium has really started using these Lithopoma snails quite a bit more, and we found out through some snail mortality, we've reached sort of like a target density that we're able to use in culture, and it does save a lot of labor time and has really increased the output of these guys. So a second chapter that Joe had is looking at the ex situ to in situ transition, right? So now we've improved our ability to grow all these little baby corals. That's great. We've got them all in these land-based tanks. What are we going to do with them? That's sort of where we were. How are we going to deal with this influx of novel genotypes? And so the aquarium had on hand a couple of different year classes, I guess we would call them, of corals. The 2018s are on top of that pyramid there. That's not the most practical coral outplanting method. Again, we're at sort of research and design scale here. So that's something that probably in the future, you're not going to be able to plant a lot of corals very fast like that. And then the 2017 year class, which had another year to grow in the land-based systems, was outplanted using a more traditional nail and cable tie method. So those were actually fragmented. They had grown so much in a year that we were able to get multiple colonies out of each coral colony. And some went straight back to the reef, and then we also partnered with the Coral Restoration Foundation and took some of them to their nursery, because the other idea here is sort of transitional phase where you bring the colonies back to the nursery, let them grow there, and in theory, we thought that would improve our survival and growth rates. We were really sort of pessimistic about the corals that went straight back to the reef, to be honest with you. We didn't expect them to do super well. In terms of where these things went, we had two different sites, Chica Rocks and Tennessee Reef, as well as the CRF nursery in Tavernier. Tennessee Reef is the more forereef, sort of historic, where you would expect this species to do well. Chica Rocks actually historically did not have very much staghorn coral. It was of interest for management reasons, I guess I'll leave it at that. In terms of survival, we're actually really pleasantly surprised. So Chica did more poorly, and this is pooled by year class. So that was primarily driven by the 2017 year class, which was the larger corals that you would expect to have had a little more of a head start and have done better. But I think just the physical, that pyramid and allowing the coral to grow in the ocean actually helped, versus we saw a lot of breakage and damage on those ones that were attached to the males. Tennessee Reef did way better than we were expecting, and COVID actually extended the sampling a little bit. That's the big gap there. We did this out to almost 500 days of monitoring. And then the nursery, as we expected, did really well at over 90% survival. And then the year classes also surprised us. So the 2018 year class, which started off as the much smaller polyp count colonies on the pyramids, ended up larger after 500 days. So that's really good news, because we now know that we can take them out of these labor-intensive ex situ systems much sooner than we thought we could. And the nursery actually was the opposite. The 2017 year class did a lot better, and that's probably because those little fragments hanging from monofilament is more typically how they're grown in the nursery. These are not representative photos. These are some of the better performing corals, just to give you a visual example of what we were looking at the end, and really how much more those nursery corals grew. I didn't point out on the past figure that the y-axis was completely different. in the nursery. So they just, as we sort of suspected, we know that corals grow a lot faster in the nursery. And then the last thing that I'm going to talk about is Diadema insularum sea urchin culture. This is this team that has been working on this. This was actually the farewell gathering for Lotus Hassan, who's in the middle. He took a job back home in Australia, but the rest of the team is there. On the right is Aaron Pilnick. He's a PhD student that's been with me since 2018. He should be finishing this semester. Marissa Moran is a biologist, and Alex Petrosino is also works for the Florida Aquarium, but is a biologist. And diadema culture is something that has really sort of taken off in interest, and a good number of people that have gotten behind it and begun to fund it. If you're not familiar, so Diadema insularum is long spine sea urchin, so critically important on coral reefs in the Caribbean. There was a big die-off in the early 80s that is still unexplained, and I think people potentially didn't realize how important they were ecologically until this die-off occurred. So the service that they provide is a grazing herbivory service that, you know, corals, even in nature, have this problem with macroalgae outcompeting them, especially in the face of eutrophication and other threats. And so Diadema provide this really important service of grazing back the macroalgae that allows the corals to thrive. So there's been a bunch of interest in restoring the species for a long time. And the sort of strategy that we've decided to pursue, given our expertise, is intensive culture in laboratory conditions and restocking, similar to what we would see with larval bivalve seeding or stock enhancement of a fish, although this of course wouldn't be a fish stock. And so this is the lab there in Apollo Beach, if you're ever able to stop by. This was pretty soon after Aaron started his PhD, and we just had an empty room that the aquarium was kind enough to let us use, as well as a bunch of resources. And there's a lot of background that went into the design of these tanks. It's actually pretty interesting from an engineering perspective, but that was Aaron's first task. And so he built the Diadema Culture Lab. And actually we've ripped out that second bank of culture units there, and we now have a settlement system because we started having some success in larval culture, and we're like, oh, now there's this issue of we need to be able to settle. So that was sort of the progression there. And this is a publication that Aaron put out last year, descriptive essentially of the system. So all the details, if you wanted to design the system yourself, are in there, as well as some of our brood stock management practices and spawning protocols. And we're happy to report, so the arrows represent, the colors of the arrows represent our ability, the reliability and scale at which we can do these things. And red actually at the time represented it had not happened yet. We have done a couple of test restockings of cultured urchins at this point, and actually there's a batch on the way down to Miami right now for partners at University of Miami to work with and take back out to the reef. So we're slowly hoping to turn these arrows green and yellow, and that, you know, that is, we're making progress. It's a really difficult culture process. It's one of the most sensitive animals you could try to culture. They just, everything kills them. Some of the research that we've done to get there was you have to culture, so they eat live microalgae of various species. And so we've done a series of experiments that were just published recently looking at microalgae quantity and type in terms of what is the best way to grow these larvae. And we've had some good results there. So not only are you culturing the urchin larvae, but you then the microalgae is almost as difficult to culture as the urchin larvae, not quite. And this is just an example. This is the most successful run to date. I think to date we've produced over 1,200 juvenile urchins that have been shared with partners for various behavioral mesocosm trials, grow out experiments, and that sort of thing. And again, this is one of the first restockings that we did in partnership with FWC down in Marathon, and that is an outplanted staghorn coral that those urchins sort of cluster into. So with that, I hope I left a little time for questions.
Yeah. Let's give a round of applause, Dr. Patterson. Thank you so much.
There's contact info and address if anybody wants to reach out.
All right, prepared. The lights are going to come back on. So Christine, you can turn on the lights here. So yes, thank you, Dr. Patterson, and thank you for your patience with our setup here. So any questions from the room for Josh?
Do you think the diadema in your snails that you used in culture are actually performing a similar task? How prevalent are those snails actually out in the wild versus just in your laboratory?
So they're really on the reef and the wild, the diadema are gonna be an important one probably. I mean, there's parrotfish and others. You really don't find those snails in high densities out on the reef in the wild. So they're kind of separate, right? The snails are really more a tool that we can use for ex situ culture. The diadema are not ideal. That's a lot of people think, oh, you can co-culture the diadema with corals, and the diadema grow too fast. And yeah. But yeah, so the snails, you find them up under bridges and stuff and in seagrass. So it's sort of interesting. They do exist, but not.
Just the question I have about sort of the future of corals is, what do you see as the actual trajectory moving forward? Is it that we are just going to have essentially reefs that we put a lot of time and effort and energy into restoring all the different components to? Or where does this whole field go essentially? From
And
a
when does it
rural
become-
trophic perspective?
And if I could just, does it gardening at some point? Like, when does
Yeah.
it become yes, when does it become self-sustaining?
Yeah, I mean, so the narrative that you hear a lot that I think people have gotten better at talking about is that this is really sort of a band-aid, right? Until we can address climate change and these other things. There are pillar coral is a good example. There are species of coral, and it got so bad with the stony coral tissue losses and stuff recently. NOAA had people just going out and grabbing species corals to be sort of a representative genetic arc in land-based systems. And so
Arc meaning like preserved, so maybe someday in another society they're outplanted, right? Like, just to put that in context, it's not like an arc, it's like Noah's Ark he's talking about.
think about what is it, like black rhinos, I mean, there are literally corals that would be functionally extinct or are functionally extinct in the wild that we would have lost that genetic diversity if it hadn't been brought into culture.
Yeah.
So that's one aspect of the tool that is helpful.
Yeah.
In terms of, large scale replanting and that sort of stuff, that's certainly one of the limitations of restoration aquaculture has often been scale. And so, when you talk about to the people that are very gung ho about this coral outplanting, we need to scale it up and make it bigger. And, there are arguments both ways for that, I guess.
Yeah.
But, there is interesting. The species profile, because it used to just be staghorn coral almost exclusively. Palmata is coming in a lot more, and so looking at trying to tailor it for specific microhabitats within the reef and that sort of thing. And I certainly don't think we're going to outplant our way out of the problems that we face, but it's a good way to maintain some level of coral cover and then the genetic diversity also.
You showed us a lot of different examples and yes, no, maybes, and particularly intrigued by these ideas of restoration aquaculture as a production system that but also improves water quality. I think that's like one of the most intriguing, exciting things. It gets people jazzed. Like, living shorelines are going to solve a lot of problems. And restoration aquaculture. So I guess we're similar to Christine's question about coral outplanting. What about this idea of restoration aquaculture for water quality improvement, especially in contrast to aquaculture, which has often a water quality degradation signature, shrimp, Atlantic
Sure.
salmon.
And that's one of the ways that even my thinking has expanded from the time I started, because I used to have this box of restoration aquaculture is you've got specific habitat or species that you're trying to restore. And it was done specifically for that purpose. One of the ideas behind the water quality improvement thing is potentially we could have clam farmers or oyster farmers that are producing shellfish for human consumption that may also, depending on how that culture is being done, may provide a water quality service. And so that workshop that I mentioned that covers permitting is also going to start talking about and having the conversations about, if you look at the Chesapeake Bay, where they have in place these payments for ecosystem services to shellfish farmers.
Right.
In practice, it's only happened a couple times. It's still not at the scale where it's supporting the industry and expanding the industry. But in theory, it is, there's a lot of data that still needs to be collected in Florida, especially because there's a lot of analogies drawn with Chesapeake Bay that may or may not hold true down here in our systems. But it's definitely a big area of interest.
It seems like it overlaps with a lot of agricultural production where there's it's already a very marginally economic activity. It's very easy to go below zero and your income above. And so if there's an ecosystem service that, even if it's, a penny per clam or whatever it is, and you start getting paid by us, by FDACs, by taxpayers to be cleaning up water, then suddenly the industry is more attractive, right, to expand. And
Yeah.
you pay the farmer, in this case, for the work of the oyster or the clam.
Absolutely, and especially, when you talk about nitrogen removal services and that sort of thing, the actual harvesting, there's the denitrification aspects, but the actual harvesting of the animal removes a lot more nitrogen usually than the denitrification aspect. So that's where I think the commercial aquaculture potentially has a really good chance to, help with that.
Well, that's easy to measure too. You just literally have it. You don't have
Yeah.
gas bubbling and all this other
Yeah.
stuff. Yeah, that's good.
You just measure the nitrogen.
Yeah. Nice.
Yeah. So
Any questions from the Zoom? I know Andrew's out there on the Zoom.
Yeah, Josh, that was a nice talk. Thanks for contributing with your presentation today. I was just kind of curious, based on your kind of read on things within the state of Florida regionally, what's your sense for kind of the priorities or kind of the funding landscape for these various systems that could benefit from this aquaculture restoration or restoration aquaculture, in terms of, you highlighted a number of different systems or kind of habitat types in your kind of quiz at the beginning of the presentation, but what's your sense for that?
yeah, it's highly varied, I think, so when you think about something like, and I didn't even touch on mitigation, right? So a lot of seagrass restoration is funded by mitigation. So that's an existing framework where if you're going to damage X acres of seagrass, which happens all the time, you are required to mitigate for or restore. That can be done in a number of ways. It can be done through protection, but also through active restoration. So that's a more established one. I have recently heard of some projects that are using corals in mitigation. I think they were looking at expanding Port Everglades, and there was going to impact some coral resources. And so that would have been some of the first sort of mitigation funding that would have gone to propagating and planting corals. That's pretty much how most of these large nonprofits that are out planting corals are funded by government grants, federal grants, state grants, private donations, and that sort of thing. But I do think, David's point about bringing up the payments for ecosystem services and how we might get some of that established is really interesting too, looking forward.
Any last questions from the room? All right, let's give Dr. Patterson another round of applause. Thank you very much. Everybody in Zoomlandia, good to see you on YouTube. We will see you next week, same time, same place. Be good.